Brought to you by Curious Moranland. So, take a look at the DNA molecule. What I want to do is demonstrate how DNA can replicate itself. I'm going to do DNA replication using models. So, let's jump down to here. If you look at this picture, it's like, all right, there's a big chromosome up here. There's your chromosome, and you can see unraveling, it coils and coils. So eventually, you can go back to the DNA. This is a double-stranded molecule. When the DNA makes a chromosome, or coils into a chromosome, you actually have two of them. This is a double-stranded chromosome. This is DNA as a single-stranded, and I actually shouldn't say stranded, I should say band. It's still the double helix. A chromosome that's duplicated because it's going to do mitosis, so this would separate, and I'll draw in a centromere later. So let's go. How do you get from here, normal metabolic activity, interphase, to doubling yourself still during your interphase, during what we call the S phase of the cell cycle, to actually doing mitosis and my hands, imagine them being a spindle fiber and pulling the chromosomes apart. So let's look at this here. Quick little review here. This is really what's going on and what I'm trying to demonstrate in here. And I don't know if the video gets all the top of this, but I mainly put this for you to see. You have a original DNA molecule, and it's going to ultimately get a new left and a right. Watch the short little animation. The color code is trying to show you that the left and the right side get a new strand. Now, this is obviously more detailed than what I'm showing here, but that's the general idea. And I want to end with something that looks like this. So I'm going to come back to this when I'm done. Alright, so let's look at it like this. Alright, so let's grab a DNA molecule. I've got a DNA strand that I've opened up. So here we go. Here's the sugar and the phosphates. Notice, if you could zoom in, you can see that these run in opposite directions. If I could do it right. Opposite directions. There's a band, like I said, double-stranded DNA. Don't confuse that with saying a double-banded chromosome or a double-stranded. This is the helix. Chromosome is DNA that's wrapped around a protein and just coils and coils and coils. If you look at that other picture. So let's do it like this. First, let's twist our ladder. We got a twisted ladder. If you're going to replicate this, you got to open this up. So there's an enzyme that does that, and it's called helicase. It kind of holds it open. I'm going to move this over here. So, and then the hydrogen bonds break. Now, our DNA is open. This is going to get a new right side. This is going to do a new left side. We often will call this the lead strand, and this the lag strand, whichever one is replicated continuously. And I need to elaborate on that later on. Now, I'm not going to do it for the whole time, but imagine doing this. So let's say here's your molecule, and we've opened it up. And then what happens is free nucleotides come in and fill it in. And then the same thing's happening here. Free nucleotides come in and fill it in. To save a little time, I'm going to just open this DNA molecule, and I'm going to the complement, the lead strand, or in this case the left side, is going to get a new right side. The right side is going to get a new left side, and now we have two new chromosomes. Or should I say two duplicated chromosomes. Now, obviously a chromosome is much larger than this. This is only 6 nucleotides long. 3 pairs here, 3. And so let's do this. I'm gonna, just so I can move these around. So we have, these are already taped together. Now, I'm going to switch to a drawing that I put in. So imagine right here would be a centromere. This is a doubled chromosome. I probably could keep narrating, but what I'm going to do is just go to this picture take a look. Over here you got your doubled chromosome. You got your centromere. That's the little blue dot. And then you can see where spindle fibers attach and attach. I don't have the little, so imagine a little centromere here. Spindle fibers attach. During anaphase of mitosis, it would pull these chromosomes. Then this would unravel, new nuclear membrane, and now we have two new cells, each with an exact copy of the genetic material. Because DNA replication is, the whole purpose is to make copies of the chromosomes. So, although it doesn't happen like this, it's like you just have one chromosome, it makes a copy of itself, but they're attached for a brief moment, and then they separate during anaphase. So that is my short little demonstration on replication of DNA molecules in what we call DNA replication. If this DNA opened up and messenger RNA came in and made like a new right side or new left side, that would be called transcription. So